Good morning, everyone. Welcome to day 2 of the presentations, Forest Genetics 2023. Hope you had a good day yesterday. Looks like a few people had a really good night, too. There's a few empty seats. All right. I don't think there's any... oh, yes, I do have one announcement to make, and that is if you are traveling to the airport later this afternoon, tonight, or tomorrow, and you have a ride, seats in your car to offer those that are also needing a ride, could you put it on the board outside? So there's a column with where ride offers, and then people that need rides can then maybe team up with you. Okay, so please do that. There are a number of people heading south either tonight or tomorrow if you're not joining us on the field tour tomorrow. There will be information about that optional field tour for the people that signed up for that later in the day, and also have some CFGA announcements to make. As you noted, we're not having a business meeting, and I'll explain that probably before lunch, before some of you start taking off. Yes. Oh, there's a survey for the Oaxaca meeting. Okay, so they have to use the QR code. Go to the site. Looks like it's June 10th, approximately, next year in Oaxaca. So they've scooped any potential dates for any other meetings in that period. So yeah, that looks like a great location for a genetics conference, doesn't it, eh? Yeah. Okay. All right. So I would like to now introduce our third plenary speaker, Dr. Laura Leites from the Penn State University. She's a research professor of quantitative forest ecology. And before most of you think she's from France, she's actually from Uruguay. So please welcome Laura.
So is this on? Good. Good morning, everyone. Very happy to be here. Has been a great couple of days. Thank you for the invitation. Great to meet so many people that I've collaborated through the years through email or exchange things, but haven't met in person. So it's really nice to see faces. So I'm going to talk today about genecology and how we can build in this foundation that they provided to us to understand responses to climate change and to guide its movement. So, 1971, we were already summarizing 200 years of studies in genecology. So by now, 250 years of genecology, ecological genetics, defined as the study of genetic changes in relation to environmental variation. And so the next few slides, I'm going to just point out to some very important findings from these studies. Many of you are very familiar with all of this, so just bear with me for a few slides. Just want to make sure that we are all on the same page. So what these studies have taught us is that in forestry species, adaptation to climate seems to be clinal, right? So we don't have very distinct populations that are separate, but a continuum gradient of populations adapted to climate. We have also seen that for species that have this population structure, these populations have different growth potential. And so here in this graph, in the x-axis, I have home climate, populations from warmer areas of the species distribution on the left, colder areas of the distribution on the right, and then some measurement of productivity or growth. And what we see is kind of that clinal variation, but also that populations have different growth potential or innate capacity to grow when they grow in favorable conditions. We've also learned that many of these populations have different climatic optimum. In this graph here, if we look at annual temperature and some measure of, in this case, height, each of these responses is one population, and what we see is that different populations within a species reach their maximum growth at different annual temperature or climate characteristics. And then that in general, growth and survival decreases when a population is moved from the climate they are supposedly adapted to climates that are different. So here in the graph, home climate being transfer distance 0, and as we move from that home climate, in general, populations decrease in growth, right, or survival. And here you have a nice example of that on a black walnut provenance trial in Pennsylvania. So the tree here on your left is the seed came from Potter's Mill, Pennsylvania, which is a couple of kilometers from the test. And you see it's doing really nicely. The signs are the same size. And this poor one came from Oklahoma, right down here, whereas the test is right there. And there's only a couple of trees left, and they look like that. Okay, another thing that these studies have taught us is that there are the presence of trade-offs among traits across a species range, right? So populations in different areas of the range are going to favor different trait values. And so here, for example, we have a graph where we have growth potential of the populations, so the innate growth capacity when they grow in mild environments, and then freezing tolerance, and we see that there's an inverse relationship. Some of the populations with high growth potential then will have low freezing tolerance and vice versa. And then the final thing I want to point out is that for some species, we have learned that the populations don't seem, some of the populations within this species reach their optimum at home climate. So again, this is height, transfer distance, 0 is home. And so for some populations, they reach their optimum, their maximum growth at home, but for some others, that growth maximum occurs in a different climate. And then finally, that we can broadly and coarsely divide the species into what we call genetic generalists and specialists based on the balance or how they accommodate spatial and temporal climate variation with species balancing, you know, let me go back there. With species having more reliance on adaptation than phenotypic plasticity and vice versa. So species C will be a species where that evidence of adaptation to climate and the populations is stronger, and so we assume that there's more reliance on adaptation compared to this one where we assume there's more reliance on phenotypic plasticity. Now, in terms of management applications, all these studies have helped us figure out seed movement for restoration or for reforestation. So just by looking at the patterns of genetic variation, then we can use those to guide seed movement, and we have done that from very early on. Okay, so the type of experiment that has been, that is kind of the gold standard for all these studies are common gardens that we call provenance trials, right? And so these provenance trials are common gardens where natural populations from the range of a species are planted together in a common environment. And so, again, sorry about that. So here we have the distribution of red maple, or all the red dots are natural populations from which we collected seed, and then the squares are our common gardens that look something like that later on. This is red oak in Indiana. And so objectives of these provenance trials, mostly the ones that were established early on, included finding populations that would improve or optimize productivity for the site, for the test site. And so that means that a lot of these tests are of regional scope, tested provenances, populations that were from around the test. Some of them were actually established to study genetic variability of a specific species. So in those cases, we do have a series of tests, and that is great because then we can quantify G by E. construct seed transfer guidelines. And then one of the interesting things here, important as we move forward, is that usually the populations tested were not too climatically distant from the test site, and so we don't see serious maladaptation because that was not of interest at that time. Now here is kind of a summary, a graphic of the establishment of these provenance trials in the United States. It's not only provenance trials, it's also tree improvement trials, but it does show you, from what I see, at least in the East, when most of these trials were established. So between the 50s and the 80s, there's a lot of these trials established, and what that means is that we have the data many times still in paper copies. We didn't have the computers the way that we have it now, and so very few of them are still in existence. And this is the black walnut trial that is still there, and I showed you a minute before. Now, these are some pictures. So I've been collecting and recovering data from provenance trials. I've also been gathering pictures that tells us a little bit about how things were when those tests were established. I particularly like this picture with that very much stylish person doing field work, very comfortable. We now have tablets, but that is really, I think, cool. I'm going to figure out who can do one of these for me. And then 1957, we have black and white, and then by 1980, we get to some color pictures. This is Kim Steiner, a professor at Penn State, that established the black walnut test that I showed you. And so this is the black walnut test when he established it. So I inherited that test, and I think it's the best gift I've ever gotten was that test, kind of my playground. So anyway, so the data we get from these tests, there are, we have to analyze, of course, and there are a few models that we have continuously used. Here we have the three of them. So, let me go back. So the first one is looking for associations between the population's performance in a common garden or more than a common garden and characteristics of the home climate. And so if we see a relationship there, association, we interpret that because it has been measured in a common garden as evidence for that adaptation to the climate or the environment the population is coming from. We also have population response functions, and here we definitely need more than 2 common gardens in which we measure the performance of a population in different tests, and so we can actually model the response of the population across the tests and the test characteristics, environmental characteristics. Now, for those of you familiar with this, realize I actually simulated data to make these illustrations. You will never have this many observations per population when you do that. So I just was, who knows what I was thinking. I think, so anyway. And then the general transfer function in which you have the performance of a single population. So each point here is one population, one garden. And so we measure the environmental transfer distance between the population and the garden. And so we have an idea of how the species is responding to changes in climate. And so early in the 90s, with a couple of papers, Chava Matias invited us to think about the provenance trials again in a different way and just realized that these were climate change experiments and that we could reanalyze just to learn a little bit more about how different species could respond to a changing climate. And so the idea then is that each of these natural populations is supposedly adapted to the home climate. We move it to the climate of the test. So we have had a climate change in space. And so as each common garden then will give us a transfer distance and a performance measure for a population. And so depending on the number of tests we have, the number of observations for the population, we can model that and kind of understand that thing. And so now these same models are allowing us to look at things a little bit different and put more emphasis on learning about responses to climate, right? So here, these ones now we can look at, okay, what are the climate drivers that are driving that association. We can look at population differences in optimum, maybe even phenotypic plasticity if we have enough observations, and then species-level responses to climate transfer. Now, one of the things as we start to reanalyze this data that came immediately obvious was that we needed to expand these models in a way that we could extrapolate some of these insights into populations or into populations that were not specifically tested in our sites, and also move beyond a species-level type of response. And so I'm just highlighting a couple of new models here that, not new because of their recent, but new after that we started reanalyzing, that allow us to do that, to kind of go beyond these models. And so by combining a couple of these original ones, we have one at all in 2010 with the model in which we do associate performance as a function of test climate and population home climate. And so now we can estimate performance for any population in any test. And then some work that we did in 2010 in which we look at population's performance across climate transfer distance. So we move from that species-level response to climate into population responses to climate, right? But the important thing that this entire body of work has done since the early 90s is to move us from that idea, move us from that idea that the species is just one unit and projecting that species as one unit into the future to a more realistic and nuanced projection of what the species response is going to be by looking at population. And so a year ago, I was in my sabbatical, and we decided, with my colleague here, Marta Benito Garzón, we decided to look at, well, how are we reanalyzing this data? What are the insights that we're gaining across the globe and across biomes? And so we did this literature review, which we selected studies that reanalyzed provenance trials data and had at least 3 provenance trials and looked at the growth, survival, and phenology. And the idea here was that we needed to make it manageable, and so we just use these 3 traits, or these 3 groups of traits. So we ended up with 147 studies, 58 species, out of which 28 had range-wide studies. The range-wide studies loosely defined because there are really no one single that we can say this is range-wide. But before I tell you a little bit about what we found, I just want to point out that we have an Excel file that goes with the paper that you can access. The information is a paper, and we have a summary of all these studies. So if you are starting to look into a species specifically, it may help you gain some time to go look at that. Now, I was in Bordeaux, and so there was wine involved, so it may have spilled in that. But I think that the information is good. So the first thing that we see is that the interest in reanalyzing these provenance trials data is really increasing. So we are recognizing the value of these trials as climate change experiments, and we are taking advantage of that. The other thing is that really most of these studies focus on regional analysis, and that has some drawbacks, because when we talk about the species, the entire range, we only have like pieces of the puzzle when we only have these regional trials. Now here is where those studies have been conducted, or at least where the trials have been located. And one of the things that you immediately see is that we don't have anything in the Southern Hemisphere, right? So these are the countries where we have trials in these either regional or range-wide studies. And so that was something very interesting. We were hoping to have at least something from Chile, but with our criteria, we couldn't find anything. The other thing is that of the species with range-wide studies, so those 28 with loosely defined range-wide studies, most of the studies are for temperate forestry species, a few for Mediterranean and boreal biomes, and then almost nothing for tropical and subtropical. And that also, of course, have implications because we don't have the knowledge level for these biomes that we have for the temperate biome. Most of the studies were conifers compared to broadleafs, and only for commercially important species. So a lot of these trials, because some of the objectives were to look for increasing productivity, they were established for commercially important species. However, there are very important species that are, from ecological standpoint, that we need to be paying attention to, and we don't have the information. So this is a summary table, and what I just want to show here is how things spread across the biomes. So as I was saying, we have temperate species is the most common, a couple, just one for tropical, subtropical, a few in Mediterranean and boreal. So of the studies that do range-wide species, so it was very kind of difficult to go. So our original intent was to kind of come up with all the conclusions from these studies, but of course the analyses are widely different and different to get information that is compatible among them. So what we did was, okay, how many studies have conclude that there is some evidence of adaptation to climate? And so here we see 79% of them conclude that for the species that they are looking at, there is some evidence, while 21% find weak or no evidence. And so what that tells us is that, reinforces the idea that for many species, responses are going to be at the population level, at least in the temperate biome, which is where we have information. Now, for the climate variables, so this one was the most difficult to try to get something, patterns out of the many studies, mostly because some studies did test different variables, but some others just use variables that other studies have found as important. But in general, for 59% of the species, the evidence that was found was using temperature-related variables. For 15% of them, precipitation, and 26% of them, both. And so one of the things that we concluded here is that we really need a more explicit evaluation of climate drivers. And the other thing that we were thinking about is kind of all of the studies look at climate normals when they are measuring differences in climate. And so I think now we have enough information to start looking at differences in climate variability and frequency of extreme events. We also looked at trade-offs among traits across the species range. And so in half of the species with range-wide studies, the conclusions of the study were that there was evidence for growth tolerance trade-off. We didn't find studies that actually looked at growth drought specifically in our summary, but in some species with range in the Mediterranean biomes or dry landscapes, there was some evidence. And so this could actually be the result of the species we are studying more than the existence of one type of trait more than another, right? So we don't have a lot on Mediterranean, for example. However, understanding these trade-offs is really critical when we talk about assisted migration and seed sourcing strategies. So that's something that we need to pay more attention to. So before I go to something else, I wanted to finish up this part just thinking a little bit about what else can we learn from these tests, right? So many of them, as I was saying, are not longer in existence, so we are kind of limited, but a few that are, can we look at things a little bit different and think more about phenotypic plasticity, which is something that is going to be so important for extant forests. We are looking, for example, at variability in annual diameter increment, looking at tree rings. And so can we use that and think about that type of measurements to have some estimates of phenotypic plasticity? Of course, phenotypic plasticity is very difficult to deal with because we need to separate adaptive from unadaptive, but can we at least start to think about these? And then associating that with characteristics of temporal climate variation the same way. And then, of course, if we do have the tests, including genomics, going to be critical for us to move ahead fastest, right? We actually just have a new colleague in the department, and we are working together. We have a lot of phenotypic data for our black walnut provenance trial, and she's sampling tissue to do genomics. So hopefully we can move things forward. Now, what are we doing at Penn State? What have we been doing the last few years? So the first thing I did when I came was, when I did my PhD at Idaho. I worked with Douglas Fair. I worked with Western Larch. I had beautiful data sets in Excel from Jerry Reffel, Barry Jagwish, all was good. I get to Penn State, start looking for data and nothing. And so I had to go on a search, and it has been wonderful. It has been really, really wonderful. But through finding, papers in proceedings and learning about trials, getting people out of retirement to help me, we have gathered a lot of data by now that came in this form and with very interesting stories behind them. We digitized, cleaned them up. At that time, coordinates were not like now that we go with the GPAs and get the coordinates. We had people that went to the trials to see if they find something for us. Completely out of retirement. We found trials just by accident. It has been a very interesting exercise. Yeah, I have actually a story for each, I think for each species except one, a fun story. But at the end, we were able to recover some data for black walnut, black cherry, red maple, northern red oak, yellow birch, and red pine, and white pine as well. So one of the things, we have to start from the very beginning with very simple questions, mostly with the broadleaf deciduous species. So we ask, is there evidence of genetic differentiation along climate gradients in these species? How much of that variation is explained by climate? And then do we see that growth cold tolerance trade-off that is so well documented in conifers in these species? And so I'm going to go very quickly through this, but what I want to show you for each species is the data we have, just because it kind of shows you the value, and also, for all of us that are still doing research, the value of archiving well our data, because you never know who will use it in the future, right? So here in red are the natural populations, and in blue, the squares here are the tests. So this is black walnut, black cherry, mostly in the commercial range of black cherry, red maple, northern red oak, and yellow birch. So it's the only species for which I don't have any nice stories to tell if you want to ask later. So one of the things we did with this data then was to say, okay, we want to look at, is there evidence of clines in growth potential? And also, is there evidence of cold-limited responses? And so what we decided to do was to divide observations. So observations where the populations were moved to milder climates were used to look at growth potential, and observations where the populations were moved to colder climates were used to look at cold limited responses. And so what we expected, if everything went well, and this is population climate, colder areas of the distribution, warmer, and so we will expect a positive relationship for growth potential and a negative relationship for cold limited responses. And so here are the results. And what we see is that for black cherry, black walnut, and red maple, we see declines. They are strong, and they are opposite as we expected. These here are the percent of variation explained after side effect is removed. And so for the three of them, we see nice patterns. But we don't see that for red oak or yellow birch, right? So we didn't find strong evidence of clines in this species. And so Preliminary analysis also indicate the Easter white pine should go to that category as well, more on the specialist type of species, whereas this one, at least with the information we have right now, it seems that there may be more generalist. Okay, so we also got some phenology data for some of these species. And so we are doing preliminary analysis. Phenology data is very interesting because it was always very subjective, many times categorical. And so, and there are a lot of interesting ways that people did things in the past. And so we are just doing, what we want is to have many species, so wherever patterns we see, we can be more confident of the results. But for black walnut, we had data for bud burst. We couldn't find any relationship between home climate and bud burst. But we did find relationships for senescence. And so here is the percent of seedlings retaining at least one leaf in October. And so what we see is what we expected. Populations from warmer areas retain more leaves later in the season compared to populations from colder climates. And as you see here, 79, that's when the experiment was conducted. And actually, the person that did conduct it, I found him, and he got me the data. It was really cool. This is red maple. And so for red maple, we did find some evidence of earliness of flushing being related to climate. Here, this was an ordinal variable, so this is ordinal regression, and so the way that you should look at is the different colors, right? So for each temperature of home climate, so this is warmer, colder, we predict the probability of the species the population flashing earlier or later, and so more the yellowish green colors means earlier, later is the darker colors. And so populations from warmer climate were flashing later on average, or had a larger probability of flashing later than the other ones. And then for fall growth cessation, we found that as we expected as well, and this is actually, it seems like textbook, and it's data from I don't know how many decades ago. It's just incredible. But so we have home climate here, bud percent, so the percent of trees that had bud set in August. And so we see a beautiful relationship there where warmer populations set bud later than the ones from colder areas. Now we are, okay, let me go back to the next one. So kind of what we are seeing right now in these two species and with the limited data we have is that there seems to be some evidence that there is association between time of growth, initiation and home climate, but not as strong as what we see in fall, in growth cessation, at least in these two. We need to analyze more species. We're going to use drones and hyperspectral cameras to look at phenology in the black walnut test. We're very excited about that. But until then, I think we are going to have to be content with this. Okay, something else we're doing is looking at early growth patterns of black walnut populations. And so the idea is, we can model height as a measure of, as a function of age. And this is very important in growth and yield models, for example. And so one of the questions we had was, well, should we move from a species level model to a model that accounts for population differences? And so we use the Chapman-Richards functions that's usually used to model height growth through age and modify it to include population climate. And yes, indeed, according to statistical metrics, we did improve the model and the predictions with population from warmer climates growing faster than population from colder climates. And so with white pine, we have a little bit of a nice amount of data, as you can see. So all in red are the populations, and then the tests are all these triangles in blue. And so we are doing a few things, but the one I wanted to show you is some work that we did with Prasad, in which we thought about the idea of the ecological and physiological optimum, where the species, the populations do well ecologically and considering competition with other species, and then in terms of their genetic patterns. And so here on the left, you have, with FIA data, we model abundance of the species, relative abundance, so using other species as well, and then the growth potential with the provenance data, just kind of looking at this idea of ecological and physiological optimum. And then we superimpose those two to find areas where both were going to be high, low, or something in between, and use that to project into the future, just as a way to aid management, to identify areas with good potential for assisted migration and good potential for conservation of genetics. We are also working on seed sourcing, delineating seed zones. This is something we did for white pine. And so the continuous variation that you find by looking at trait in common gardens, we then categorize it into the seed zones. This is still work in progress, because one of the things that we find out is that when you do these zones, we were using a classical approach where you put a confidence interval around the population mean to delineate the zones. And what we saw was that the confidence level that you use really impacts the number of zones and the width of those zones. And so here is mean annual temperature for two different confidence levels, and the red bars indicate how wide, not only the number of seed zones, but how wide they were. And so you see that usually we go to lower confidence levels just to lower the probability of type 2 error, but really increases the number of seed zones and makes them very narrow. So what we are doing is trying other things. So we've tried unsupervised cluster analysis, and we are just trying to move away, but we haven't just quite yet find a solution. So where are we up now in the future? One of the things that is happening right now in the US, at least in the Northeast, that we have a big need for seed zones for different species and seed sourcing strategies for a changing climate. And so we have recovered data for more species through collaboration with Dana Nelson in the southern states of the US. So we have now thousand pines. We have collected more traits for some of these species. And so using some of the models developed and that we plan to do simulation experiments to try to come up with climate-adjusted seed sourcing strategies. So the idea being that if we have a site to plant here today, can we do climate-adjusted provenance by combining seed that is adapted to the present climate and then future climate, but decreasing the quantity of the seed as we move into the future climate, right? So in theory, this looks great, but we don't have any information on how to actually do this. So our hope starting this fall is to start working with simulation experiments and see what we come up with. So with that, I want to go to acknowledgements. Of course, the biggest thanks is to all the people that have done provenance trials over the decades. Jerry Reffel, who has been an amazing mentor and got me in the path of ecological genetics. The students in the lab, so all of these students have contributed to all of this work through time. None of them are right now in the lab, only Josh. So, but they all have worked on these things. And just because we only have one student, we're going to be recruiting. So just if you allow me here, if you're interested or know someone that may be interested, please help me and spread the word. So with that, thank you, and take questions.
Thank you, Laura. That was a beautiful talk, and that paper, your review paper, is really lovely. So everyone here should read it. I'm curious. So you found that there's more evidence for adaptation to temperature than to precipitation and precipitation combined, although those were there. And I always wonder, and we find the same thing with our Western conifers generally in seedling trials. And I'm curious if you think that that's because of it has any effect of the scale of sampling, since water availability varies spatially so much. There's so much greater, smaller scale variability in water availability than in temperature, or if you've thought about that. And the other question would be, did you look at species range size? I can't remember that. To see whether there was a relationship between whether or not there was climate adaptation and range size.
Yeah. So to your first question, so one of the things with the precipitation variables, temperature variables, it was really difficult to find patterns, right? Because it seems there was some studies really did look at differences and tested different variables, and they came up with temperature, but many use temperature variables because someone else have found. And so those numbers have to be taken into that account. Whether that's an issue of scale, I haven't thought about it. It could definitely be. I do think it's also probably the biomes where you were studying that could have an effect, that if we go and look more Mediterranean type of species, or species that have more xeric distributions could become more important. And then to your second question was? The size of the range. Oh, the size of the range. So that was something that we really want to do. And actually, this was all modified by COVID, and we ended up working for 4 months. But my hope was to look at the range, and I still want to look at characteristics of the range in terms of climate variability, right? How we quantify the climate variability in the range, and how can we associate that with what we see in terms of the strength of adaptation. I think that would be really cool. With my 5 broadleaf species, we have 3 that have strong evidence, 2 that not. I look for commonalities among the 5 of them, up and down, and I couldn't find things to kind of explain why. But I think it would be really cool to do that. And now we have a lot of species. Now that we know where they are, it may be feasible, right? But we try. It was, yeah.
I think I noticed that you don't have the trials in the southern hemisphere, and I know conifer is not native into the southern hemisphere. They are just introduced species in there, but I think the CAMCOR program at the North Carolina State University has trials in the southern hemisphere, maybe in South America and maybe South Africa and beyond. So have you looked into those? And the reason I'm asking about this is because for much of the world, the forestry is based on conifers, which are not native species. So you may have provenance trial and progeny trials in there that are based on where all the populations have been introduced. So they may have meaningful data, at least to show you what the species can take, even though you will not be able to ascertain local adaptation because all that is growing there is not local. So have you looked into that? And the second one is, why do you want to create seed zones for eastern white pine? Why the general trend is to move away from those fixed zone system of seed management in forestation?
Yeah. So to your first question, there may be trials in the south. We just don't see them in the literature, right? And hopefully there will be people working on that soon. To your second question, we do a lot of natural regeneration in the east, and we are just starting to, and so we don't plant a lot. And so the culture of planting is still not there. And so we are trying to start with these fixed zones first, just to get to the idea of seed zones, and then because it's easier to work with. So that's kind of the main reason there. Yeah. And you had. Yeah, that's the one I just answered. Yeah.
Laura. Hello. Oh, Laura, thank you for for a great presentation. Do you have any advice for organizations that who are establishing new provenance trials, particularly perhaps organizations with limited budgets?
Yeah, yeah. So I do think we have a lot of information in these old papers, right? You have to go to proceedings, but that's one of the things I also would love to have a student do, is to just go through all that and gather all the information we have about the common gardens, how they establish them, how they evaluate them, how they manage them, because I do think that can save us a lot of grief as we try to reinvent the wheel now. So I think that would be really great. The other thing that I think is that we are losing a lot of knowledge with people that have done these things, are not longer working, they're retired, and mostly in the East. Like people were always super happy to give me their data, they went out of their way, but I've never talked to anyone, right? And it seems to me that we definitely need to kind of bring them together. At some point before COVID, we were planning on a workshop, trying to get these people out of retirement and teach us, how did you do it? What are the things we need to be thinking about? And I think if we do that, we will have a lot of good information to kind of proceed in a more and smarter way, right? Not start from zero and try to do things that haven't done before. Yeah. Okay?
Thank you. Okay. On behalf of the committee, we'd like to thank Laura for presenting today. It was a wonderful talk.
Thank you.
A small gift for our appreciation. Thank you very much.
Thank you. Thanks.
I'd like to welcome up Ms. Song, who will be our moderator for the next session. Thank you.
All right. So welcome to session 5, seed transfer and provenance test analysis. I'm appreciated to be here as a moderator for this session. And my name is Hamann, and currently I'm a postdoc research fellow at University of Toronto. And it's my honor to introduce our first speaker. So our old friend, Dr. Brian Cullis, who will introduce us the other R package, which named dwreml.
Thank you. I'll be up soon. Oh, yep. That's slide 2, but that's okay. Thank you for the introduction, and thank you again for the opportunity to talk again. I shouldn't be doing this talk, obviously. It should be Dave Butler. Because I think I mentioned in my first presentation, Dave wasn't able to come, but most of the good work is due to Dave, and anything that's going wrong with the package is probably due to me. So you'll have a chance to really have a go at me at the end of the talk if you like, which I'm looking forward to. I did miss my first slide. Anyway, I'll go back. Okay. That's all right. So dwreml, it's an R package for feeding the linear mixed model. There's myself, Alison and Dave. And yeah, we'll just give you an update as to where we are with it and what we're aiming to do. So the motivation for the work, or for the project, is sort of outlined in this slide. I'll go through it quickly because I know we're short of time, so I don't want to make things difficult for anybody after me. 25 years since the core of dwreml was devised, maybe longer actually, early 90s. It's really stood the test of time quite remarkably well, due to Robin Thompson and Arthur Gilmore's amazing capacity to implement this thing called the average information algorithm. Also, it's got such a huge range of models which include, for modelling the variant structures, which include genetic ratedness. It also then took another leap forward when Dave came on board and myself, and we moved the moved the unfortunate poor user away from standalone dwreml into a slightly better computing environment, which is the R environment. However, lately it's been starting to struggle quite a lot. It's running out of gas. There's been a huge demand back home for fitting models with genetic relatedness, particularly on markers, and that's really had a really serious impact on the ability of ASRL-R to fit what I would consider simple models even to small data sets in real time. We work closely with a lot of plant breeders back home, and the turnaround time for their analyses is very short. They're in a very narrow window, and ASRL just sits and chugs and chugs and chugs and chugs. And so it's rather frustrating for the breeders. In fact, it's very frustrating for the breeders. And unfortunately, there's a tendency to move away back home and elsewhere to people doing what I think is piecemeal, approximate multi-stage approaches, which is really not what I'd like to see happen. We want to strongly encourage best practice, which is single-step type stuff. Okay, so the background. So what we want is an open source R package, which fits the linear mixed model and estimates variance components by residual maximum likelihood. The sort of specs we want, I think, but you can add a few more or take a few off, because we're very, very keen to get user feedback. An ASRL like R functional user interface, efficient ordering, analysis, factorisation and solution of mixed model equations. That's the big cruncher for ASRL. For anybody who probably hasn't done maths for quite a while, basically in high school you're solving simultaneous equations, and ASRL's big job at the start of the AI algorithm is to solve a massive set of equations. And as your number of equations gets bigger and bigger, and as those equations become more dense, which means there's filling, then ASRL dies a slow death. So we need to sort that out. We also want to put it into a modern computing environment, which is C++, rather than Fortran 90. We want to at least match what ASRL can do in terms of variance models, but we also want to put new ones in, and we want to do it, and we want the users to be able to interact with that. We want to also address problems with conversions, because that's a really ongoing issue with ASRL, as you'll see later in this talk, and hopefully we'll get this stuff out into the public domain. So this project started under the guise of dwreml, which could be called dependent Wollongong residual maximum likelihood, which it really isn't, but I'm not allowed to tell you the true reason for the acronym because Robin would kill me. Robin being my mentor, Robin Thompson being one of the most clever men I've ever met, or have met, probably the most clever man. It's myself, Dave, Alison, I've also got Luke on board. And in this talk, that's what we'll try and do. Okay, we'll try and give you a brief overview of the components, give a little insight into the core, summarise some of the variance models, illustrate some of the calls for some tree breeding examples, and finish off with a bang, I hope, to show you how, well, I think we've done pretty well in sort of 18 months. Okay, so the features that we've got thus far is we have got an ASRL front end. That's great because URW sort of owns the IP. We do have a fantastic way of solving the mixed model equations using this French-based sparse direct solver. It's all been written in C++. There are lots of other companion packages to do lots of cute things, and we will go to the public domain. Okay, not wanting to do too much maths, but just give you a little flavour. So direct methods for solving the mixed model equations require some form of this thing called Gaussian elimination, which is just simultaneous equation of solving equations like you did in high school. There are much smarter ways of doing it these days. This Cholesky factorisation of this coefficient matrix is what we basically use. And rather than doing one line at a time, you can smartly order the equations in C so that you can access multiple similar rows of that matrix and solve them not one by one, but actually in chunks, which are called supernodal chunks. And these modern solvers are fantastic because they're also parallelizable. Okay. A very, very quick overview of the dwreml core for those of you who are using ASRL-R. you'll probably think, well, it's almost the same, which it, not surprisingly, would be, since Dave Butler is the author of both packages. So why would Dave change something that seems to be working pretty well? So it's got the standard look to it. So ASReml would be there. It's just DW there. You can you could just take your scripts from ASReml and almost use the same. Okay, there are some changes, and it's got all the hooks that you want. You can do multi-site analyses by doing diag_site.genotype. You can do residual dsum and all that sort of stuff. So it's all pretty good. And we've got pretty much every model that you would like to fit that's in ASReml is currently in dwreml. I won't go through them. It's a bit geeky and a bit boring. That one's a bit geeky and a bit boring. I'll skip that one. We don't want to bore you with that. Okay, this is better. Now, Trevor's been kind enough, or silly enough, to let me and Alison loose on some data of his. I'm sure he'll never buy me a beer again, but that's all right. So Douglas fir example, model environment trial, 47 progeny trials in 39 environments, which is a point of interest, which I could explain, but I don't have time. 247,000 trees from 1876 parents. Sadly, the parental fill-in was well below what you'd like, but it's not easy, okay? And I'll come back to that. That 13% is a killer, all right? So there's here's a few lines of code we create. We're just going to do a single environment at the moment, okay? Just do it easy. So we just do some R code. There's only 4,000 lines of data here. There's only 150 females and 19 males, and some reps and some rep.sets. Okay, cool. Well, here's our dwreml call to do that, which looks just like ASReml, pretty much. So height, modelled by the mean. Our random terms are rep and rep.set. And then we've got this cool thing here to use a relationship matrix, A star, and we add a half mum plus a half dad. Kind of cute stuff. Dave, I'm very proud of Dave for that. It's very, very clever to do that. It's kind of contorted, but it's very, very, very useful in many applications. And the residual doesn't have spatial variation here, but obviously one can do that. Okay, some timing comparisons. As I said, most of our original motivation comes from the people we work with back home, which are grain crop breeders, annual inbred crops. Chickpea is an example of one of those. This is an example. It's got 46 trials across 2 years, 29 environments, which are combinations of location and year, 4,200 odd varieties with data, 4,900 varieties with pedigree records. So we've got a few which don't have, which aren't, didn't have pedigrees or something, I think. 26,000 records with data. 13.7% fill-in of varieties, which is low, but much better connectivity at the parental level. So what we did is we did a meta-analysis, everything's single step. Single-step meta-analysis involved sequential fitting of 4 linear mixed models to the individual plot yield data. Sorry about the feedback, I'm not sure why. And what we did is we fitted factor analytic linear mixed models to the additive variety by environment effects, going from an FA1 up to a 2, to a 3, to a 4. And we maintain an FA1 for the non-additive variety by environment effects because we're in the rather unique position of being able to fit models to our non-additive effects, and we standardly fit a different factor model to additive and non-additive, which kills ASReml. But you'll see it doesn't really hamper dwreml. We also fit autoregressive spatial correlation models within each trial, and we've got a whole swag of random effects going on in the background for other things. All right, this is a stunning slide, in my humble opinion. At convergence, the two packages basically reach the same likelihood, almost identical, same parameter estimates, which is kind of cool. It would have been a bit disturbing if we didn't, obviously. I wouldn't be here at the moment if we didn't, probably. We start with FA1, and what we do here is we do FA1, one iteration. For those people who've got the misfortune of using ASReml, iteration 1 always takes a long time. It sits, and sits. And then from there on, it gets a bit quicker. Doesn't always converge, as you know, but at least you'll get something. Okay, so we have 3 columns here of times, seconds per iteration for FA1, FA2, 3, 4, okay. dwreml is amazing, in my humble opinion. It just blew me away when I saw how clever Dave is. Here we got ASReml, 3,300 seconds in, in like the first iteration and 600 and something in the second, and so it goes on. And as you get the complexity going on, that number just keeps stepping up, and we couldn't even get an FA4. Just ran out of memory. Okay, couldn't even get close. VSN have just released 4.2, and so we thought, well, why not show some numbers for 4.2? They've done very, very well. I have to say they've really improved 4.1, but still dwreml, even after 18 months, and we haven't fine-tuned it anywhere near as fast as we can get, we're still pretty good, I have to say. All the way down there, we're tracking about 10 times faster at this point, which is pretty good, because hopefully we'll go well beyond that as we go. I'll finish with this last slide, which is incredibly busy. I don't know how much longer I've got, but I don't want to do any more after this. This will be enough, I hope. This is back to Trevor's example, and thanks so much to Alison for putting all this together. So it's basically similar to before. We've got all sorts of different models for modelling the additive by environment, or additive parent by environment effects. And so, similar to before, we've got 2 extra models at the top, which is what we call the diagonal model, whereby we've got different genetic variances at each site, but no correlation. Core H is another model. So basically, here we go. All right, this is the scary bit, because dwreml is a beta tested package at the moment, under test after 18 months. Right, ASReml has been out there for a long, long time, and this is the scary thing, because yeah, fine, we start with default start values to fit this model, and they're both identical. We do a model here which is core H, and they're both identical. We start from default start values, right? And there's no way that a user can provide ASReml with sensible start values, really. You can, but most users, I'm afraid, wouldn't, and you get issues with convergence. ASReml says it's converged when it's not. And that really annoys the crap out of me, because I feel really bad for anybody who hits that and doesn't realise that they do, because you will think you've converged, but you haven't. Okay, that's a really, really horrible problem, and we're very, very much aware of it. So dwreml got to a much higher likelihood from default start values, which was kind of impressive and kind of pleasing. When we give the 2 packages some more informed start values, fortunately, the 2 packages converge to the same number, which happens to be the same number as that. Having said that, we're not there yet, because when we do an FA2 from an FA1 start, there's small differences there, and I know why. We know why that's causing it, but the differences are small. FA3 start from an FA2 start, that means we take the variance parameters from an FA2 model, we then plug them in as starting points for the FA3, which is what you should be doing, I think, as a user at the moment. And even then, ASReml converges to a likelihood which is incorrect. Well, certainly I don't believe it's the right likelihood. That's a much more sensible likelihood. And then FA4 from FA3, it gets worse. So the take-home message there is user beware at this point in time. There are issues. I think we're getting towards something whereby we're solving them, but I think we've got a lot of work to do in that space, particularly for complex models, or the more useful models like these factor models, which are the ones that I think we should be using, particularly to inform ourselves of G by E in a more sensible framework, so we can address some of the things that Laura spoke about in a more formal and a more comprehensive, statistically efficient manner. And that will probably do. I don't want to overrun my time. Thank you.
Wonderful presentation, perfect timing. Any questions for Brian?
Thank you. Brian, Brian, can you explain a little bit better what parental fill means and why is 13% a problem? I don't understand that too clearly.
No worries, yeah. Parental infill is basically connectivity, if you like. How many parents are in common between your different experiments or environments. So the example that I presented in my first talk was where every mum and dad or every family was present in every environment, so that would have 100% infill. That's the perfect world. Sadly, when you're dealing with putting together historical data sets, like most of the trials that we deal with, certainly in the case where you're pulling together progeny trials or provenance trials or these home gardens or whatever, you very rarely have good fill-in. And that's where fitting these models become really, really challenging. Yeah, computationally challenging, getting it to converge, so forth and so on. So I hope that helps a bit. I should mention also, oh, sorry, yep.
So one of the problems that I've run into with ASReml in the past is the inability of the model to do both spatial analysis and sort of, multivariate type analyses where you're, doing bivariate, multivariate, MET, whatever,
sure,
with a spatial residual.
Yep.
Any hope for that?
Well, I believe yes, because, where are we? Which one is it? I think it's this one. Yeah. Okay, it's that one. So chickpeas, we do spatial analysis, is almost ubiquitous in Australia. AR1, yeah. And so, which is where, I think let's say that on that slide there. Yeah. So, so the reason why ASReml is being flogged in timings here is partly because of the fact that there's two FA terms going on, which really gives it a hernia. And the other reason is because you've got spatial correlation going on, which means you increase the complexity of the equations. And so ASReml dies a terrible death, okay, because of that. And that's part of the reason that you're probably having here. The other reason that you're probably struggling, I'm not familiar with your data, but you haven't got any true replication. We have the inbred crops has the ability to have true statistical replication, obviously, but that's not necessarily the reason. That's probably more to do with this one, with the computational load that ASReml's got to go through, whereas dwreml doesn't seem to be having the same problems. There's a fellow in the back there. Towel. Sorry. Shouldn't be doing that, should I? It's someone else's job. Oops. Towel.
Thank you, Brian. So I was just wondering, what is ASReml 4.2 doing differently to improve itself so much?
Oh, look, I can go through the geeky stuff if you want. Like, Arthur's finally realised that there are better algorithms out there for sort of factorisation and reordering the equations, because if you order the equations in a smart way, this is geeky stuff we can talk about later. But basically, Arthur's realised that there's some things he can be doing better, which we've been trying to encourage him to do for many, many years, but he's been resistant. So that's good to see. But as you can see, the French solver is phenomenal. Luke's PhD compared a whole range of geeky ways of solving the mixed model equations, and Mumps came out second behind Luke's own software. But Luke is humble enough to say, well, mine's only PhD level. Let's use the Mump solver at the moment, which I kind of gave him a big hug. Because I think that's really a big problem with a lot of people who are IT people. They tend to like to squirrel away with their own software. But no, Dave's really good like that, and so's Luke. Yeah. Anyway, I should get off, I reckon.
Thank you, Brian.
Thank you.
Lovely presentation. All right, so next speaker, Dr. Andreas Hamann from University of Alberta, who's also my PhD supervisor and gave me a lot of help about research. And his topic is balancing risk and the benefits of assisted migration to address climate change. Welcome.
Can you hear me? Am I amplified? Yeah, it sounds like it. Okay, good. Do I have to kick this off? Or, oh, there we are. Okay. So I will talk about assisted migration, risks and benefits. And I like to start by asking, where are we in terms of climate change? So for me, personally, this is a bit of a milestone or a benchmark. I got about 20 years ago started with Tong Li and Sally Aiken's lab on developing those climate software packages. And we made predictions for the 2020s at that time, and it seemed like a long time ahead. But here we are, it's the 2020s. That is very interesting, so we can be held to account for our research at that time. The other milestone is that we completed climate normal. That's always exciting for us. So the 1991 to 2020 average, it always takes a while until all the weather station data is in. But that's how climate is defined. So climate is defined as usually as an average 30-year period, and we can compare it against the previous normal, the 1961 to 1990 benchmark. So this is what this graph is. And so now we really know what the climate is or was in the 2000s. So the midpoint of this period is 2005, when we got started. So if you want to know what the climate is now, you have to invite me back in 15 years. And if I can still make it up here, and the youngsters let me, I would be able to tell you. So we have about a 1 degree warming in British Columbia and Alberta on average, a little bit less in the south, a little bit more in the north, relative to the previous climate average. There's a shortcut, though, to figure out what the climate is now, and that's future climate predictions. And there's also a milestone. CMIP 6 has been released, so this is the latest set of multimodal climate predictions. They're also now all integrated into our software packages. And we can look up what the climate is right now. So it's very little uncertainty for this, because so we're zooming into Alberta here. So the top row are the absolute climate values, and those all mean annual temperature. The bottom here is the relative changes. So we like to set the 61 to 90 period as the reference, and then you can see we got about a 1 degree increase for the recent normal. We are right here in the middle, so 1.5. That's what, if you plug in a 2020 prediction, they all give you the same answer because half the data is already in. So it's about 1.5 where we stand right now. If your reference is pre-industrial, we are about at 2 degrees. So 2 degrees is where things become a little bit uncomfortable. And we've seen signs of this. So there's quite a few papers on, so I'm now focused mostly on Alberta, so aspen dieback, white spruce growth decline, mountain pine beetle epidemic. So these are all partially attributable to about 1.5 to 2 degree warming relative to pre-industrial times. And of course, we have fires. Fire frequency appears to have increased. So this is where I live, right there. And it was in 2017. We didn't even have a fire nearby. These were fires that were burning more than 1,000 kilometers away in BC. So the new meme is, we don't forget the Anthropocene. We live in the Pyrocene in Canada now. So this is and we can keep this going. So everything that burned down is only about 2%. So we can do this every year for 50 years. So even the undergraduate student will have retired by then when this is over. Now, let's talk about the context of assisted migration. So I think it also highlights how urgent this becomes. So I start again with a 1961 to 90 normal, and this is a climate envelope, essentially, calculation for white spruce. So the white area is where white spruce is, where we do forestry with white spruce. So So you can translate this into a multi-dimensional plots. So this is basically the dashed line here is the climate envelope of white spruce with the frequency indicated by those colors. And then I give you a couple of surrounding ecoregions as reference. So up here it gets warmer and drier. Down here these are the alpine regions, so it's cold and wet. And you can see, so this is our past climate. This is the 2020s, so midpoint 2005. And this is where we are right now. So these are the model predictions. There's a couple of them for the 2020s. Those are the circles. Very little uncertainty. But we have shifted our climate envelopes already quite substantially. So if you think about this being white spruce forestry area, we shifted that, or this envelope has shifted quite a bit. So it would be wise to catch up and move seed sources accordingly. So to guide this, we have a couple of simple tools. I like to work with those. Climate envelope modeling, common garden experiments. For risk assessment, I like to look at historical biology too and at landscape climatology. So that's what I'm going over in the next slides. I think climate envelope matching is still the 101. If you want low risk assisted migration prescriptions, keep the climate constant. So you try to match some source environments, so whatever you've done here in forestry, you might do over here or over there, depending on what the best climate match is. This should be generally relatively safe recommendations. So if you have a good climate match, that's definitely a good thing. We've done all this work 10 years ago, but we're now updating it with the new climate normals and the new climate predictions there. So we have this done for spruce, but there are also older papers that have done it at an earlier time, what the correct climate matches be. Common garden trials, that is the 201, I think, of supporting assisted migration. It's a little bit more sophisticated. We are now looking at growth as well, not just whether it's a climate match, but whether we get satisfactory growth. But alone by itself, it can involve a little bit of risk. But nevertheless, I think transfer functions beautifully explained and response functions, universal transfer functions. So we heard a very nice summary by Laura this morning, and also throughout the conference, this got mentioned many times. So this will allow us to pick provenances that are actually optimally adapted, that have the best growth, best survival. That's another piece of the puzzle. So I wouldn't say it's the only piece of the puzzle, but that's definitely a second important criteria for guiding which provenances to plant. And I also want to point out that you don't necessarily have to model. So for Alberta, we have a lot of trials, very high connectivity. These are our test sites here, the triangles. These are our provenances summarized, so the things that got moved. For example, here in the red zone, we tested a lot of local red sources, but also sources from all other breeding regions were tested there. So if you have high connectivity, you can basically just calculate averages. And plus, we don't have to simulate climate change because we already have experienced it. So if you just calculate averages of those transfers, you get essentially recommendations of what works in terms of growth performance and what doesn't. So it still needs a mixed model because the connectivity is not perfect, but these are sort of very robust estimates of what you can do. So not every movement north works. For example, if you move from the wetter foothills to the drier upper boreal forest, that does not work. So red means growth declines relative to the locals, but the northwest transfers, they generally work. Any kind of moving things south, I mean, we could test it, but you wouldn't do that under climate change, but clearly these things don't work. This kind of information you also want to consider when providing assisted migration guidelines. One of the problems with building those response functions is often that you don't have sites at the extreme ends. So we used dendrochronology data because while you don't necessarily have a dry site, there will be a dry year at a reasonably dry site where you see where growth collapses. So you can use this kind of information to tie down response functions and get slightly better predictions of growth that are completely just modeled by climate, and you can apply climate change and see where productivity peaks for different provenances that way. So those are our, I think this climate matching and growth performance modeling, those are the two most important things. But I also like to assess risks that are associated with this. And so we found in the last couple of years that historical biology approaches through tree-ring analysis can be quite insightful for this, as well as landscape climatology. So one of the projects that gained quite a bit of insight was the lodgepole pine experiments, the big Illingworth trial. Many of those trials got killed by mountain pine beetle, and we could take them down and look very carefully at the tree ring records and anything that may be hidden in there. And so David Montoy and Miriam Isaac and Renton did this work for that provenance trial. And so there is some insight that growth is not everything. So one of the things, if a provenance is happy where it grows, this is how it looks like. So you get clean rings, no issues, late wood, early wood. But if you move things too far north, then you get late frost, late spring frost rings. So the meristem get damaged, and it leaves a trace of this. Other things that can happen if you don't match the growing season quite right. So if the tree still grows in the fall, you get these blue rings here, a lignification of the late wood doesn't take place anymore. I don't know if that poses a real problem, but it is an indicator that the growing season doesn't match with the available environment. So these things tell us which provenances, while they may grow fine, they are not 100% comfortable. And so this was followed up by a bunch of students. It turns out that there's all kinds of other anomalies, white rings, double rings, isotope analysis you can obviously do to see how well the water efficiency compares between provenances. So there's a lot of information you can tease out whether the provenances are actually feeling comfortable where they grow. So that's early warning signs. So it tells you, while this provenance is still doing okay, lately we get a lot of anomalies. They don't really like those environments anymore. It's time to move. On the other hand, it also tells you if you move too far. So if you push it too far, they also become uncomfortable. They leave all these anomalies in the wood. The last piece of the puzzle is landscape climatology. So not looking at biological data at all, but simply at climate in the landscape. And you'll hear a talk by Sang about this later in the morning or afternoon, I forgot. So Sang looked at frost risks of late spring frost that are purely due to landscape position. So if you move sources, you might move them inadvertently to environments that have very different, relative to the triggers where bud break and end of growing season is triggered. You may have different risk environments, and this is purely because of climatology patterns in the landscape. So you can inadvertently, accidentally increase the risk more than you think. Even though the average climate match is still okay, the extremes don't cooperate. And then the last one is velocity. So forward velocity is a metric that basically tracks how fast species have to move, or populations have to move through the landscape in order to keep track of climate change, to maintain constant climate conditions. And you can also do this backwards. So if you're asking, I want to reforest here, where would I have to source seed to get genes that are approximately adapted? It can be a long distance. So in that case, assisted migration would have a high priority, or it may be just sourced down the slope if you are in a landscape position that allows upward movement. So that would be a low priority for assisted migration. Sang, how many minutes do you have left? 1.5, okay. Let me just skip to a few slides. So one of the things that we want to try is to balance risks and benefits. So the way you can do it, let me skip through this, is through an online tool that we make available just for exploration, so where you can set climate matches and growth factors and risk factors. So I recorded this as a video. I hope this works. So if you go to the website, it provides you with some instruction. You can pick a breeding region where you would like to reforest. So that is the blue marker there. Red means they don't grow very well. Green means these are sources that grow very well at this particular location or in this particular area. So you can set your criteria, what kind of growth you like. So if you look at the poorly performing provinces, they usually come from high elevations from the mountains, just as Laura also predicted this, summarized it this morning. So you can set this to something like 90%. I want to maintain 90% of my growth. And now we can set the climate match. So if I set the climate match as good for the 1970s, it restricts the provenances that I can use. But I can also do this for the 2030s climate now and say, okay, I want that climate to match for the 2030s. And you see you get a slight northward movement into the seed zone or into this breeding region. You can set additional criteria such as only northward movements allowed, and other risk criteria are going to be included into this as well. So as you do this, you get seedlot suggestions from the Alberta Seed Bank or from the available seedlots. So if you set your criteria too small, nothing is left. So that's an important piece. You know, we would like to avoid risk, but if there's nothing, no options left, that's also a problem. So that potentially allows us to set the criteria correctly for making the best reforestation recommendations that we can make. And I think that's it. Thanks for the attention, and thanks to all the people who contributed to this.
Pretty cool video. Any questions for Andreas?
Thanks very much for that. That was super interesting. I love the work that you're doing out for Alberta there. So in the figure that you showed, looking at the shift in range of white spruce in Alberta climatically, what it's showing is that the predicted climate for the 2020s is basically already moving from the highland where, you know, white spruce is a dominant well-growing species into parkland, where it is mixed wood, lower proportion, poor growing. So have we seen a lot of indication that those areas are moving to that? I know that there's the paper in 2018 showing the decline in white spruce, but since then, has there been any additional indication of poor growth in those regions?
Yeah, it's, you know, we also do experimental studies with white spruce quite a bit with my colleague Uwe Hacke, who's a tree physiologist. White spruce has the ability to hang in there very, very strongly. It does not immediately die, even if it gets pushed beyond its envelope, but the growth declines quite dramatically, and there is really a very sudden drop where everything breaks, right? So it's a very strong threshold in terms of tree-water relationship where it very steeply declines. So I think we haven't really seen that dieback in spruce like we've seen it in aspen. Aspen is far more gradually. So aspen is happy to die back and then resprout from the roots. It has a different strategy. But to answer that directly, no. So there is no indication that spruce is about to go out at the southern fringe, right? Yeah. Although there is some more work from Saskatchewan, from a professor there. I forgot his name there, but he did demonstrate some of these things. Yeah. Yes.
Thank you very much for the nice talk. I thought the tree ring data was really cool, and I was just wondering about connecting that to Laura's talk. You said you were able to collect the tree rings because there was the whole provenance trial was killed by the mountain pine beetle. Do you think as more provenance trials are decommissioned or, put out of service, there's potential to collect a lot of more tree ring data and do this in many other places?
I think if I were to collect tree ring data, I would not go anywhere else but to provenance trials, right? This is so much more informative than in situ, because there you have your environments totally confounded with genotypes. So there's very little you can learn from tree ring analysis in natural stands. The key really is to go into provenance trials, old provenance trials, especially the really old ones that were planted in the 1960s. So that's good. That's a super resource. So yeah, I definitely would do that. Good point.
Sorry, we are kind of running out of time, but if you have any question, you can always talk to him. All right, thank you again. All right, so our third speaker, Dr. Cuauhtémoc Sáenz-Romero, and his presentation is about establishing Monarch Butterfly overwintering sites for the future.
Thank you very much. I thanks Greg for inviting me and sponsoring me to be here, otherwise it will be difficult. And I'm going to talk about monarchs, butterflies, and trees. Why monarchs go to the overwintering sites in Mexico? Because under the crown of the Abies religiosa, sacred fir, they have a very specific microhabitat, enough cold to do not spend the fat reserves to fly back to Texas to put the eggs for the next generation, but not too cold to get frozen if there is a rain in the winter. So Abies religiosa has already a very narrow climatic space, and the monarchs use a subset of that small, narrow microclimate under the crown. Oops. How can I come back? So with Jerry Reffeld, who has been also my mentor, and we modeled the suitable climatic habitat of Abies religiosa, and the red pixels are the suitable climatic habitat for the end of the century. This is the Monarch Butterfly Biosphere Reserve, and you can see that by the end of the century, it will not be a single square kilometer of suitable climatic habitat inside the reserve. That means that we need to migrate, in my view, Abies religiosa to a higher altitude volcano. This is one of the options, Nevado de Toluca, or Popocatépetl Iztaccíhuatl. So that is a complicated possibility, and it's what we have been suggested. We have done these altitudinal bands based on common garden tests with this technique that was developed by Jerry Reffeld to estimate how much of difference in altitude make two populations genetically different for quantitative traits. And we suggest move upwards. The problem is, what do we do here? Because there is no more altitudes inside the reserve. The reserve, Monarch Butterfly Reserve, has about 3,550 meters of maximum summits. That is the problem. So we have done what we call protected assisted migration, which means moving to higher altitude the provenances, but in the case of Abies, it's very important to use the shade protection of shrubs. And this is the survival under the shrubs, Baccharis conferta in this case, and this is the survival in the out in the open field. So it's a huge difference. So we need a companion to do the assisted migration. And So what are our options? One is Nevado de Toluca that has a summit at 4,700. Abies religiosa reach a maximum around here, 3,400 meters. So we tested at higher altitudes up to the timberline. One thing is to plan that, model that in the computer, publish that, and one very different thing in Mexico is to do it. So Ben, what should I do? I just click. So I have a 4-minute video for you. Should I click again or? Okay. So I have to credit this video that was made for 3 students, undergrads of my university. I don't know even how to edit a video, but these guys are terrible. So, thank you. This is the effect of the climatic transfer function, of the climatic transfer distance. 20 years ago, I came to Vernon for first time, and I went to the field with Barry Jacquish, who showed me a preliminary transfer function, and it was a site in a very poor location in terms of development of the fitness. And I said, why did you plant it there? And the answer of Barry Jacques was, because I like to know where is the zero. So Barry, it took me 20 years to find the zero, and it's about here. So no growth at 5 degrees of movement. Even is a negative growth because the seedlings have not growth in 2 years and they have some damage. So it's very, it's a lot impact, the movement. And yesterday in the field trip, Greg told us that you are thinking to move about 3 degrees. So 3 degrees will be about half of what will be the local population. So it's a large impact, but we may have alive trees as we can, as we can see, what I'm doing, as we can see in the survival. So if we move 3 degrees, we may have something like 60% of survival. And the seedlings are suffering a lot, especially at the higher sites. It seems to me that what is happening is that at night it still happen temperatures below zero. But what has changed is the pattern of rain, where the rainy season, which usually is end of May to November, is now shorter. So, for example, last June, we have no precipitation and a heat wave, and the rainy season is ending 1 month early. So dry season is like almost 2 months larger. So even at 4,000 meters, now the soil is very dry at the end of the season. And at night, temperatures are below zero, obviously, at 4,000 meters. So it's like the seedlings cannot handle that combination of very cold nights and the soil already too dry. It's very unusual that. So in Mexico, we have a lot of types of vegetation. And of course, we have tropical dry forests. And tropical dry forest has a very dry season. But the trees that are there drop the leaves. But it's not the case at 4,000 meters in the timberline. So when Jerry Reffel invited us to do the 2012 paper of the biomes of North America, he introduced the concept of climate without analogous contemporary, which means a climate that we cannot associate at the present with a biome because it doesn't exist. So as many things that Reffel says, I didn't understood completely what he was saying, but it took me 10 years to understand that this is this. So a combination that doesn't exist and for which the seedlings are not adapted. So when some ecologists challenge my view and say, oh, you shouldn't do this assisted migration because Abies religiosa is to become an invasive species, I don't see this poor seedling becoming an invasive species. So I thank the sponsors, and I hope there is questions. How much time do I have?
You still have enough time for all the questions.
One thing more, no matter how many papers do we publish in what impact factor are, the important thing is to convince these local actors to do the forest management differently. Yes, I don't know. Who has the microphone? Okay.
Thanks for the great talk. That was really interesting. So I don't know how faithful monarchs are to returning to the same overwintering site, but do you anticipate that the monarchs will be able to find a new site naturally, or will you also have to move the adult butterflies during their overwintering flight?
Yes, I get this question frequently. I work with trees. I am not specialist really in monarchs, but my understanding, well, in these other volcanoes that I show, including Nevado de Toluca, there are small overwintering sites, and some of them relatively new, very small colonies. So it seems to me that monarchs have the plasticity to change of overwintering site, but I don't know if the shift will be in enough population size numbers to do a viable population. That I don't know.
Thank you.
We are trying to do something different, and let's see what happens. Yes.
Thank you so much, not only for the work, but also the video and the outreach and the applications you're doing. My question is actually more about the video and the outreach to local peoples. So you said the video was made just by some undergraduates?
Undergraduate students, yes.
Yeah, can you tell us more about the development of the video, and then who do you show the video to? And yes, how else are you explaining the experiment to local peoples?
Well, we have found that these YouTube videos are very powerful tool to do extension, because we don't have time nor money to be doing talks in schools or something like that. And as I said, it's a general issue. It's a generation issue that my young students are fully skilled to handle this and me not. And the music was composed especially for the video for one of these students. So we hope to do more in the future.
So the results you showed from the effects of open versus companion planting were very interesting. And in many of our field trials, we find some severe issues with competition from establishing species on our sites, specifically grasses, hampering conifer establishment. And so I was wondering if you had any idea whether the effect you saw was more of a microclimate effect or a reduction in that herbaceous vegetation under the crown, so to speak, of the shrub.
Well, we removed some of the grass to avoid competition, and we have measurements of the microclimate under the shade of the shrub. And what happened is that it reduced the extreme temperatures, either at night or during the day, because at 4,000 meters you have a very strong insulation, so during the day the sun hit hard. So it's mostly an issue of reduction of the extremes, and I believe that this idea of do not use roofs comes from the development of forestry focusing in timber production of pines, where pines are, as you know, intolerant to shade, but Abies is very tolerant to shade, so there is no a negative effect of competition at all. We have a paper on for frontiers in ecology and evolution about this issue. So it's good to have another people from Latin America.
Hello, thank you again for the presentation. My question is, how do you go about battling the climate without contemporary similar climates? Is there a way, what are you doing to help, for example, with the dry soil? Do you have irrigation, or do you just...
There is no way to do irrigation at that altitude, and we are pushing to start the production of nurse plants in the nursery. We have a production experimental. If you go to the Monarch Butterfly Biosphere Reserve, there are many NGOs that are producing seedlings to do reforestation, because it's a nice thing to do and it's okay. But nobody, absolutely nobody, produces roofs in the nurseries. They produce either pines or sacred fir. So we are testing ways to propagate, because there is no culture of propagation of the roofs. So if you ask when is ready the seed, they say very vague, ¿cómo se dice vago? Vague answers, because there is no culture. So we believe that the way that reforestation is done needs to be changed, and you know, the campesinos, the peasants there, they know that shrubs are good, but there is not the culture to produce that in the nursery. And we found also that these shrubs are very good, but the one that we have at 3,800 or 4,800, which is 4,000 meters, excuse me, is senecio, and the crown is very poor. So now we realize that we need to migrate to some of the shrubs that works better for protection, like Baccharis conferta.
Gracias.
You are welcome. De nada.
All right. Okay. Wonderful presentation. I love the video. All right. So last but not least, so our last speaker, Samuel Grubinger from UBC, and his presentation is about multispectral vegetation indices from drone remote sensing quantify clinal adaptation to climate.
Okay, thanks. Hello, everyone. Thanks for having me. As mentioned, I'm in the Integrated Remote Sensing Studio at UBC, so not a forest geneticist, under the supervision of Dr. Nicholas Coops and working closely with Greg O'Neill. So big thanks to Greg for making this PhD happen. First of all, so this work took place over the ancestral lands of at least 12 different First Nations. So in lieu of a local land acknowledgement, I just want to express my support for the land back movement. We work within this very top-down system in BC, and I think the first step to true collaboration is giving land and resources back to Indigenous people. I'm going to frame this talk through 3 different lenses. The first, methodological. I know most of you are not remote sensing scientists and not familiar with it, so I'll talk about the technologies that we use and some of the workflow to actually get from drones in the sky to phenotypic data. I'll then give some results. Some of them are operational, about the actual applications for existing trials, and then others are newer, about new patterns that we're seeing with this data. And this was just published last week in Global Change Biology, so I'm going to give an abbreviated version, but there's more detail there for those interested. The trials I'm working with are Greg O'Neill's interior spruce genecology and climate change provenance tests. Very cutting-edge trials. When planted in 2005, there's a huge range of genetic material represented, from New Mexico and Arizona in the south to the Northwest Territories to Haida Gwaii to Ontario. So big differences, and also many orchard seed lots in addition to wild stand seed. There were 17 sites planted originally. I sampled 6 of them, the ones that I could drive to. So I did a climatic gradient from a coastal Mediterranean maritime climate in Vancouver Island through continental climates in the interior to a subarctic climate in central Alberta. So while I didn't capture most of the geographic variation, I did get most of the climate variation in this series. However, warming is slightly overrepresented. I don't have any site where the majority of populations are being tested in a colder climate than their provenance of origin. And these sites look really different from each other, despite being the same trial design. We have Harrison. This is a coastal site near Vancouver. Huge trees, rapid growth, high crown competition. Skimikin, your classic Christmas tree farm, pretty perfect. And then Kalamalka, that's at the site that we toured yesterday. The majority of trees are dead, and those that are alive are mostly not looking great. So this is a really great opportunity to test some of these technologies too. We have many different conditions represented. Most of this hinges on a technology called digital aerial photogrammetry, or DAP. Many of you may be familiar with LiDAR. This is not that, but it gives you a similar kind of product. So you get a point cloud in 3-dimensional space where each point is analogous to a pixel that has been located in multiple images that have been aligned. So it's the same principle as air photo interpretation, stereo view, but iterated over millions of times through a software. And from that 3D point cloud data, you can produce structural metrics. So this is one class of metric. Some of them are similar to field assessments, like height. So here we have height percentiles. It's a different way to measure height, but it gives you something similar. So this is either the height under which 99% of the crown occurs, or the height under which 999 per mil of the crown occurs. So these are our height proxies. And then you can generate more novel metrics as well. This is the apex angle, so the average angle of points relative to the leader, a measure of conicality and crown form, and then convex hull or concave hull volumes as well. The second technology is multispectral imagery. So this is a separate flight. We fly twice when we go out. And it's a camera-like sensor, but it gives you 10 distinct bands rather than a red, green, and a blue close to each other. You get narrow bands at targeted parts of the electromagnetic spectrum that are supposedly physiologically meaningful. And from this data, we produce multispectral indices. So these are ratios, mostly normalized ratios of values that are correlated with something of interest. So the normalized red edge index is associated with stress in conifers, related to greenness, the proportion of woody vegetation, while the chlorophyll carotenoid index is two targeted values that capture that shift from a green to a yellow in the visible, and that's highly correlated to the ratio of chlorophylls and carotenoids. I'm going to briefly go through my phenotyping workflow here. So it all starts, so on the top we have structural metrics, and on the bottom is for spectral indices. So it starts with a canopy height model, where each pixel represents the height above the ground of a portion of anything. And then I generate a grid from row column census data and snap each tree point with its identity to the local maximum of that point cloud. And from there, we can apply an automated crown delineation, in this case, a watershed delineation. This works really, really well if there's good space between the crowns. If there isn't, it requires some manual adjustment, which is a big labor need in this project. And then we segment the top 50% of each crown to control for occlusion and differences in physiology in lower branches. Some of these have even been trimmed, and that's where and then we clip that portion of each point cloud, and that's where we actually derive the structural metrics. For spectral information, it's really important to use sunlit pixels. They're more spectrally stable, and in conifers, that's kind of the physiology that we're trying to get at. So we use the position of the sun, and again, this 3D height map to project the sunlight and produce shadow masks. So we get the proportion of direct versus scattered irradiance from the drone. We use that to produce two different maps of illumination, average them correctly, and then ditch the worst pixels, essentially. It's an arbitrary cutoff, but I eliminated the worst third of pixels. You will lose trees this way, especially if you have big height differences and a north-south trial layout, which we usually do. But we try to keep the most pixels we can without sacrificing data quality. And then we average the usable pixels according to those indices to produce our spectral traits. So some results. The first thing forest geneticists always ask me is, can you give me height? Can you measure my trees? And the answer is yes, at least at the genetic level. So here we have BLUPs, so each population genetic value for ground measured height versus our ground predicted height. It's a very simple model using that 999th per mil height plus a lag adjustment because there was a year difference between measurements at some of my sites. Volume is trickier in DBH. We can't measure it, but we can estimate it, we can model it. So in this case, it's mostly driven by crown volume. The crown volume is very closely correlated to DBH, but adding additional remotely sensed indices to that model significantly improves the result. So crown volume plus height plus some spectral indices of your choosing gives you a good and highly transferable estimate of height. And these are cross-validation results, so tested with models either naive to the population or naive to the site, and performance is good. So to look at these variations in relation to climate, I produced these clines, transfer functions. I conducted a Pca, and using the climate Western North America annual variables, and you get a pretty clean temperature and moisture component. So I looked at transfer distances along those two principal components at each site, and you get a result like this. You can then do that for all sites, for both PCs, for all traits, and you start to get a bigger picture of clinal variation across the whole series, across all populations. So note that not all sites have both positive and negative transfer distances represented. All populations are experiencing warming at that site. So you don't have both sides of zero. Bear with me, there's a lot on this slide, but if we look at kind of the big picture, we start to see some patterns, right? So we've got ground assessed DBH in height, structural metrics from remote sensing, and spectral indices, and we have two different principal component transfer distances. The x-axis is the absolute value of that clinal relationship, either in a positive or a negative transfer. So the cline in a warming direction or the cline in a cooling direction. So some interesting results, first of all, and this still shocks me, that we see significant clinal variation in spectra at all sites. And so that tells us that there's probably an adaptive basis to some of these measurements. And also surprising, we see very weak variation along those temperature transfer distances for both field assessed height and structural data. So the difference in tree size is not reflecting temperature as much as it is moisture, at least at these interior sites, right? At coastal sites, so these are Vancouver Island and a site near Vancouver in the mountains, warm, wet, mild sites, and we see the opposite pattern, right? We see, first of all, very strong clines for spectral information, and second of all, relatively weaker clines for tree structure along moisture gradients versus along temperature gradients. So I don't know exactly what this means yet, and I'm kind of hoping that you all will tell me, but it, I think, is good evidence that productivity is not necessarily adaptation, that there's more to adaptation than stem growth. I like to show this image. This is from the Harrison site. Big, big tree, nice straight stem, great growth, but it's not doing so well. There's something else going on there. And that's what we're picking up with some of these remotely sensed metrics. I think it has something to do with moisture versus temperature limitation. You have a switch from the coast to the interior where moisture becomes limiting, hotter, drier summers. Pests and pathogens I've conveniently ignored so far, but they're there. And weevil, especially at these sites, is prolific. And then needle retention and resource allocation. We know that there's more going on at the needle level, the stomatal level, that doesn't necessarily pan out in differences in growth. And I think we're getting at some of that. So the next step, I think, is phenology. I think that's where we're going to start to tease apart some of these relationships. I have done a longitudinal study at one of these sites, at Skimikin, where I went back 9 times over 3 years, winter, early summer, midsummer, late summer, to look at changes. So these are 3 trees that are actually next to each other in the trial, and we see they react very differently seasonally. So we definitely see strong patterns there. I have yet to tease them apart, but I think that's the next step. So take homes, remote sensing is really cool. You should hire me for postdoc. Operationally, we can measure the things that we've been measuring, the old traits, height and dBH, but we're also seeing new patterns that we haven't been measuring before, at least not efficiently, operationally. And these patterns are especially evident at sites simulating a warmer and wetter future, and along temperature transfer distances. So that's pretty relevant for climate change, I think. Big thanks to Fiber, our industry partner, and NSERC for funding this, and again to Greg for making this PhD happen. Here's my contact. Thank you very much.
Hey, Sam. Thanks, Sam. So I'm going to play devil's advocate for a second. Okay, so you show convincingly that you can find correlations between these spectral phenotypes and provenance. Are those spectral phenotypes related to fitness in the trees at all?
Are they related to fitness?
Yeah, because that would have to be true for local adaptation to be there, right?
Right. I don't know. I mean, I guess that gets at how do you measure fitness in these trials, right? We don't know.
Thanks so much. That was a really impressive talk. I'm guessing that the answer to this question is like orders of magnitude, but can you tell us how much more efficient, cost-effective it is using the drone versus using people on the ground?
Yeah, that's a big question. So in terms of time in the field, it's efficient. These are 20-minute flights as opposed to a couple days measuring trees, but you still have to get there. And as John has pointed out to me, if you're going to pay someone to go out to the bush, you have to do it, right? These are remote sites, and we still have to get out there. And we have a lot of kind of weather and lighting condition issues to deal with that you don't if you're just measuring trees. You have to fly pretty much at solar noon within a couple hours, and changing lighting conditions make it difficult. So that's one piece. It also depends what you want to get out of it. If you just want tree heights and you can slap a point and clip out that CHM for each point, that can be quick and dirty and you'll get the same results. If you want good crowns that aren't overlapping to get spectral indices, that is more work, it is more labor, but that labor is in the lab and can be done by whomever, whenever, as opposed to in the field on a tight schedule by a contractor. So I wouldn't say it's more efficient, I would say it's differently efficient.
Very good talk, Samuel. My question is, are you considering using thermal cameras along with LiDAR and your multispectral?
Yeah, so Olivia presented on Tuesday. There is a thermal camera that our lab has, and we are collecting data with it. How useful it will be is to be determined. It's messy data, right? Every time a cloud passes overhead, the temperature of the foliage changes. And it's a sensor that's designed for construction work as opposed to our kind of surveys. Stay tuned. We have it, and there are plans to analyze it. It would be fantastic to be able to get reliable temperature data from these foliage, but there's so much variation, and it's temperature is emitted rather than reflected, so it's much messier in that way.
All right, so thank you for the presentation, and thank you everyone for the participation. Is there any pronunciation?
Okay, just before the break, I just wanted to acknowledge some people, especially the program committee, for putting together such a wonderful scientific program here for us. So when I call your name, please stand up so we can all clap for you. In particular, Greg O'Neill and Brian Baltunis, who kind of took the lead here for us. Okay. Alvin Yanchuk was on the committee. Sally Aiken, too. Barb Thomas. Barb, are you here? There she is in the back corner. Thanks, Barb. And then a couple of people that couldn't be here today, Nicolas Faux and Miriam Isaac Renton from the CFS. So that was our committee. They did a great job. Thanks. Okay, we're a little bit behind schedule, but let's reconvene for our next session, which is a concurrent session at 10:30 sharp. Does that make sense? Yeah, 10:30. Okay.